These are your notes on protein synthesis. Make sure that you have your note sheet as you're taking this either at home or in class, and this is part of your level 4 grade, the making proteins. So make sure you have all this filled in. If you look at some of the background information, it's actually your proteins that determine your traits. That's a very important concept. We say that DNA is the genetic code. Anytime you have a code, the code codes for something. And so it codes for, or your DNA codes for your proteins. And it's your proteins that actually go and build your structures and determine your traits. And if we look at some of the other structures involved in this, the ribosomes that are in the cytoplasm are responsible for making proteins. And ribosomes make proteins by chaining together amino acids. And this is something that or this idea of ribosomes and amino acids and proteins um, was introduced at the very beginning of the year. In Unit 2, the biochemistry, proteins were one of your organic molecules, and amino acids are what make up proteins. The word ribosome should be familiar. We talked about that in Unit 3 with cell parts. That's the site where proteins are actually made. We chain together amino acids. When we think about this and how this relates to us, um, we think of food. If you can just think to yourself what kinds of food um, are proteins, um, we think of things like meat and peanuts and almonds and chicken, steak. Those kinds of things are proteins that are food. But if we look at something like a hamburger, that's generally cow protein. And your body's challenge is when you eat like a cheeseburger or a steak, is to turn that cow protein into human protein. And so it does that by digesting. It breaks apart that cow protein, which is just a bunch of amino acids, and then whatever your DNA is, your code, puts together those amino acids in a way that makes sense for you personally as a human to then go ahead and make your own human protein. And so that's what this section is really all about. How do we get from the food that we eat, the protein in the food that we eat, to human protein? And that's what this whole process of protein synthesis is all about. Going back to one of the other structures, again, we say DNA is in the nucleus. It determines what proteins are to be made. That's the genetic code. And so the question is, if the plan, let me move this box over here, if the plan for making the proteins is inside the nucleus, and we know that proteins are made in the ribosomes, the question is, and again, if I'm going too fast, just stop it or pause it or go back. The question is, how do we get the code that's in the nucleus to the ribosomes where the proteins are made? That's the overlying question. When we want to make our proteins from the food that we eat, we want to turn that chicken protein or that cow protein into human protein. How do we get that code to the ribosomes so we know which amino acids from the food that we eat need to be lined up? And the answer is RNA. This is where RNA comes into play. Ribonucleic acid. RNA transfers it. Before we go into the whole process of the steps, you need to be familiar with three types of RNA. The first kind is ribosomal RNA, rRNA. And this type of RNA makes ribosomes. That's what the R stands for, ribosomal rRNA. The second kind is messenger RNA, mRNA. And mRNA it's single-stranded. Remember, DNA is double-stranded, and has sets of 3 bases called codons that code for specific amino acids. So before, I'm going to back up just a little bit so you can get a little sense. If it's not going to let me write, that's okay. But if I'll just continue on, and then at the very end we can talk about the differences between RNA and DNA. But if we look back again, single-stranded DNA has 2 sides, RNA has one side. That's what single-stranded means. And the bases in RNA, back up a little bit, are grouped. If you have one long strand of RNA, it's grouped in a group of 3. I'm going to make sure my mic is still on. It is. All right, so we see 3 bases grouped in 3 called a codon. And that code, that 3-base code, is going to code for a specific amino acid. For instance, if this 3-base code was in place, it would code for the amino acid methionine. Right? Now, there's a difference here with RNA. Right? There should be a big difference. This is called uracil. There's no thymine or T in RNA. Right? There's a U or a uracil. We continue the strand here. We got another 3-base code that codes for leucine, another 3-base code that codes for instance, arginine. You don't have to know all these amino acids. You would be given a chart. But you do need to know that the messenger RNA takes the code from the DNA to the ribosomes. And that messenger RNA is divided or grouped into 3-base codes that code for amino acid. The third kind is the transfer RNA. Transfer RNA, it's single-stranded, just like messenger RNA. And it's folded over on itself. So if you take a sheet of paper and just fold it up, that's what RNA or transfer RNA looks like. The bottom part, if you take a sheet of paper and just fold the 2 ends up, the bottom part has a 3-base code to it called an anticodon. It's going to match up with the codon eventually. And the top part is going to be carrying amino acid. That amino acid comes from the food that you eat. So the transfer RNA, its job is really, it's like a tow truck. Right? Its job is to, once your food has been digested and broken up into pieces, it's going to go pick up those amino acids from like the chicken or the steak that you eat. It's going to tow it to the ribosomes where you can start to begin to put those amino acids in a special order to make your human protein. And again, that anticodon is a sequence of 3 bases that match the codon of the messenger RNA. What I would do again is, if I'm going too fast, which I probably am, and you need more time to write, just pause this as you're going through it. So here's your single-stranded, where's my mouse? There it is. You've got your single-stranded RNA, right, divided into 3 base codes. You don't see any T's again. You see a U because this is RNA, and you just fold it up, just like that. In the bottom, you have a 3 base code, and at the top, you have an amino acid that comes from the food that you eat. That's transfer RNA. Those 3 base codes at the other end make the anticodon. We're all set. So those are the 3 types of RNA, right? If you want to compare RNA to DNA, right, we can do that at the very end, actually. So here you have some examples of transfer RNAs with different amino acids from the food that you eat, and the anticodon, which is going to code. It's a code that's going to match up with the codon of the messenger RNA, which you'll see in a minute. That this is going to make our human protein. So if we look at how we make human protein synthesis. All right, the first step, it occurs in 2 steps, transcription and translation. You must transcribe a code, and then you must translate it. So transcription is making messenger RNA. Again, this is how we're going to get that code from the DNA to the ribosomes. So if we look at the first step, right, the first step, just like in DNA replication, since we have to get the code from DNA, DNA must unzip. That's the key word, DNA unzips. I'll give you a second to write this. The second step, the bases pair up. Now we talk about RNA bases pair up with the code in the DNA. And again, uracil is going to replace thymine in RNA. All right, I'm going to move on. If you need more time, just pause it. So here we have our DNA in the middle here. It's going to unzip, and these free-floating, I'll do that again, these free-floating RNA molecules, nucleotides, are going to match up with the base pairs in the DNA. Just like, it looks just like DNA replication. The issue is on the outside is the DNA, and on the inside is the messenger RNA. It's going to take that code, it's going to transcribe or copy that code from the DNA, and once it's copied, it's going to be released from the DNA and go to out of the nucleus into the ribosomes. It's going to release, just like that. The DNA comes back together, and now it's going to go out of the nucleus and again to the ribosomes. Out of the nucleus to the ribosomes. Out of the nucleus to the ribosome. So now we have the code here. We have the code, which amino acids, we have the code to tell us which amino acids should be lined up in order to make our protein, our human protein. So the final step is translation. How do we translate that code? Right? In the first step, the codon of the messenger RNA matches up with the anticodon of the transfer RNA. That's waiting there. Again, the transfer RNA, its job, it's like a tow truck or a taxi cab or a bus or something. It's going to, its main purpose is to go get the amino acids from the food that you eat and tow it to the ribosomes where the messenger RNA is there, so we can match up, or so we can get those amino acids in the right order to make our human protein. And why do you need to make protein? Well, it helps build muscle. It helps make enzymes, which make every single chemical reaction work. So now, again, our whole job is to line up the transfer RNA molecules in order with their amino acids to make our protein. So, messenger RNA comes to the ribosomes. This is the code from the DNA. Remember, 3 base codes. Right, this is like a puzzle. So the transfer RNA lines up with the messenger RNA, and it tows its amino acid. We have another transfer RNA that lines up, matches up with the messenger RNA. We have another one that comes. So it's all happening at the ribosomes. If you look at the pairing, A pairs with U, A pairs with U, C pairs with G, C pairs with G. So now we have everything lined up, and once they're lined up, the amino acids are brought close enough together to form a bond. So you get a bond that forms, just like that. And the ribosome is kind of like a wheel. It's gonna move these transfer RNAs out. Right? And the bond's gonna form, the amino acids are gonna detach from the transfer RNAs, and they're gonna form a bond here. And those transfer RNAs are gonna go pick up more amino acids and bring them back. I'm gonna move it back here so you can write this if you need more time. Again, anytime we're doing this, just pause it, stop it, so you can continue to write. And here our bonds form. Now that chaining together of amino acids forms the protein. Now our protein is complete. It's released from the transfer RNAs and available to turn into an enzyme, a structural protein, and we're all set. So that is how you get human protein from the food that you eat. If you have any questions with this, please let Mr. Matthews know. We will be going over in class. It's really important to understand the differences between DNA and RNA, and we'll cover that in class. But please make sure you have these notes finished. This will be part of for those that are doing the level system for this particular unit, this will be level 4.